And on to our next speaker here, Dr. Alessandro Prigione. Please go ahead and take the reins. Looks like the screen share is initiating, but not seeing any video, not seeing your presentation yet. Oh, here we go. Also, your audio, please unmute yourself, Alessandro.
Yeah, so do you see my side? Hi, everybody. Thank you very much for kind invitation. Sorry, do you see my screen?
Yes, we hear you too.
Okay. I don't see it somehow, but I can start. Sorry, I don't know what's happening.
We can see your screen. So
Okay.
if you just advance the slides, even just manually with on the keyboard.
Okay, one sec. Sorry, let me try it again. Okay.
We see it now, and then I think.
Okay, perfect. Yeah, sorry, everything's fine. Yeah, thank you so much. I'm very excited to be here and also following all these great lineup of speakers. So yes, especially because in our lab we are mainly focusing on understanding and Leigh syndrome and hopefully finding therapy. So also my whole lab is connected to follow this great conference today. And also we're thankful, of course, for Casey and CureMito ATP6 also for the recent funding. So I'm very excited to be here. So yeah, so basically our work is focused on using induced pluripotent stem cells. We just heard a great talk from Jessica before, and we use them to make brain organoids. You see a picture of these sort of brain organoids, and these are they were called mini brain because because they're small and they sort of model a brain. But now this name is discontinued because they're not actually a small brain, but they are they have some features of the brain, but they're not reproduce exactly what the brain is doing. So how are they made? So first we start, like Jessica already mentioned, we start from somatic cells. For example, in this case, we can start from fibroblast. And then we reprogram to the pluripotent stem cells, and then we can differentiate to any kind of cells that we want. So in particular, brain organoids, which are these sort of aggregate of cells that contain cells of the brain. So like neurons, neuroprogenitors, astrocyte, oligodendrocyte, which are collectively known as glia cells. And then with these cells, they're not only just there, but they form organized structure. That's why it's resembling the brain. And there these canals to understand the interaction between different components, can understand specific aspects of the brain or of diseases. What is still a bit challenging are the fact that another component of the brain, microglia, which is important for the inflammatory response, is not automatically there, so it needs to be exogenously added, and also blood vasculature, which also is important for the inflammatory component or also for the response to environment of nutrients. So this is also exogenously added. So this means that it's an imperfect system, but can allow you to identify dysfunction that are primary starting in the neuronal or glial cells. So this is, for example, is a picture, and you can appreciate that there's an organization. So you see here in green, these are progenitors. They organize in this neurogenic zone or embryonic-like ventricles. You can also see sort of layerings in the cortex. So there are things that can be investigated with this model. And you can look at layer formation, neuroglia interaction, region specificity. So you can make different region of the brain. And like I said, there are some challenges, but a lot of people are working on trying to improve this challenge related to vasculature myelination, because oligodendrocyte which makes myelin, which is this part that it's wrapping around neuronal axons. These can appear very late, so these parts are still, let's say, in construction. So how do we use this model, and what do we want to achieve? So yeah, so like I said, in our lab we are mainly interested in Leigh syndrome. So we heard a lot, so I'm not gonna repeat. Just let me just stress again that is recognized there's some specificity of the defect, like we heard the talk from Shamina, from Simon, that there's some specific defect in the basal ganglia, striatum, or midbrain. So this region seems to show some specific degeneration. We're approaching that by splitting approach, like if we follow what Vamsi was saying, so that we are generating model of a specific genetic condition. So in particular, we have a model for SURF1 and for UFS4 now as well, and also for ATP6, which is also known as a maternally inherited Leigh syndrome. So and today I'm going to talk only about these two. So basically, for SURF1 is a nuclear gene, like I said, so that it can be approached. Well, the model that Jessica described, we can make isogenic lines. It means we can correct the mutation with CRISPR-Cas, and that's what we did. So if we generate control iPS cells, we can introduce the mutation, and from patient iPS cells, we can correct the mutations. So in this way, we can identify the specific consequence of the mutation and not of any other things that could be in the background of other genes or other confounding effect. And so we can see the SURF1 is a assembly factor of complex 4. So complex 4 activity, COX, is affected when there's mutation, whether it's in a patient iPS cells or in a control iPS cells. The important thing, the mutation has to be there. So this is to show that it's a precise model. So with this model, now we started to make these brain organoids, and we started with a more general organoid approach, which is also was defined as cerebral organoid, which is basically not region specific, but in general how the brain would form. So what we found is that when we had brain organoids carrying SURF1 mutation, whether in a control background or in a patient background, they were smaller compared to the control organoids. And also when we look at neurons, so because this is what we can look in these organoids, we see in the control organoids that at day 40, because you need to let them grow for a long time, right? So at day 40, there are in the control organoids, neurons are developing, and day 90, you see even more. And we see in the SURF1 organoids, this is completely disrupted. So the neurons are either not being generated or degenerated. And we can also confirm functionality that when we try to probe the functional activity of the neurons, so from these organoids, we can also grow the neurons in 2D, right? Like was also Jessica was showing, you can grow cells also in normal plates. And you can see that in the control neurons, you can see nice spiking, so the neurons can be active. But in the patient neurons, this is disrupted, and there's less spiking and less postsynaptic currents. So, but now we have this model, and then we can start to understand, like I said, is it the neurons were fine and then they start to degenerate, or they were not fine in the first place? So we look at the component of the neuroprogenitors. These they are the precursor that then give rise to neurons. And so here you see in a control organoid, is in red are the neurons, in green are the progenitors that I mentioned from these nice neurogenic areas that are organized. And also here they're very nicely organized. And in red, these are apical progenitors versus proliferative progenitors. And this again is disrupted in these organoids, so that there are progenitors you see in green, but they are not properly forming this architecture. So what we think is that this defect that we see in the neurons could come also from somehow an inability to make this neuron correctly. So we're trying now to go more into the details or to understand whether this is happening in some specific regions. So we started making midbrain organoids, which are organoids for a region of the midbrain, and they have to express a specific marker, which is TH, tyrosine hydroxylase, which is a marker of dopaminergic neurons that reside in the midbrain. And what Carmen found is that in the control organoids, which are also bigger, we see a nice development of neurons and also of their branching. But in the patient, in the mutant midbrain organoids, we see that not only they're smaller, but also the ability of the neurons to make this projection and branching is impaired. So this seems also to be true for this specific region of the midbrain. And then we can start to understand now how this region can speak one to another. So Stephanie is making different region, cortical and striatal organoids, to assemble them. So you can try to see how they speak to each other. You can start to first make the cortical organoids and maybe label them with GFP, and then see how these labeled neurons communicate to the striatum to understand whether these projections are impaired and then would impair the communication. And then we see another experiment with a specific AAV targeting neurons, so that can we first apply to cortical, then assemble them, and then look basically that the neurons can go to the striatum. And now we want to understand what happens when such a SORFA mutation or Leigh syndrome mutation would be present, whether this communication might be affected. So, but this altogether makes a sort of the hypothesis that when we start from pluripotent stem cells and we go to neurons, there's basically a very different demand of energy. And the cells need to adapt to that. And they adapt to that by switching to oxidative phosphorylation. So basically to mitochondrial respiration. And this is what happens in a normal scenario. There's this metabolic programming that at the level of progenitor cells, they switch to oxidative phosphorylation. When we're in a situation where this is not the case, for example, for Leigh syndrome or like I said, for SORF1 mutation in particular, these neuroprogenitor cells might not do what they're supposed to do and might not properly commit to neurons, and these neurons might not form the proper branching. So by looking at this data, we hypothesize that there are some components in Leigh syndrome that might resemble a neurodevelopmental disease. That means there's some defects that are underlying in the neuronal generation. And then, of course, there might be situation like we heard also from the case described for first by Leigh, that the patient might seem normal or might seem only smaller. So the children, they seem to be developing normally, but they are maybe on the lower curve of development. And then there's something that triggers something to really that cannot be go back anymore to the previous state. And we think that there's some underlying defect in how neurons are made in their let's say ability to overcome challenges, for example, inflammation or an increased need of energy. And then when this challenge arrives, these progenitors and these neurons are not able to face this, and then sort of this the degeneration occurs. So, but if this is the case, can we start to target these MPCs and finding therapies? Because at the end of the day, that's what we want. Okay, we want to understand disease, but at the end, we want treatments. So we previously looked at these progenitors carrying ATP6 mutation for meals, and we found that they had mitochondrial membrane potential defect, which would then cause dysfunction of calcium homeostasis. So now we're using this as a screening target because we think that progenitors might be important and then might be important in driving the generation of neurons. So if we can find something that improve the progenitor's function, that could be important as a treatment. So we started now a new consortium called Cure Meals with several partners from Europe, and we are looking basically into this. So we have developed now several ATP6 iPS cells carrying different mutations from different patients, and we make these progenitors, and there we are carrying a large compound screening with the help of Ole Plesser from Fraunhofer Hamburg, where they have all the automated center. And we're looking at drugs that are already either approved or safe in the clinic, but not approved for any entity. So these are sort of repurposable drugs. It's a number of drugs that can be used for another condition. And while this is ongoing, and we'll have several hits, but we already have one more advanced. And with this, everything started with one boy that is carry a TP6 mutation that had basically this sign of Leigh syndrome and had very severe metabolic crisis. And then our clinical collaborator, Markus Schulke in Berlin, decided to use this FDA approved that we identify in these NPCs carrying ATP6 mutation. So since there's no animal model or not yet, and this drug is safe in children, he had a compassionate study. And this went very well. Then this boy recovered in terms of strength, ability to breathe. And so, of course, this can be anything. We heard that also from Bamzit, when we heard about the curve of the disease, there's some spontaneous recovery, especially for mtDNA mutations. So we now have actually three, but then there's another one, 4 additional compassionate studies in Germany, and then one is in Italy. And all these seems to have improvement in the mainly basically in the preventing this further metabolic decompensation. So we see also some improve in one case of the walking distance or the fine motoric movement, but in general, the patient had peripheral neuropathy. This is still there. What we see mostly is in this part related to endurance and also in sort of helping this compensation, and we're thinking that this could act through some calcium mediated mechanisms that we are exploring. So with that, I conclude by summarizing that we think that using brain organoids from iPS cells, we can hopefully make new model system for Leigh syndrome to couple with available one, like for the one example, NDFS4 knockout, or the one that Michael mentioned with ATP6. And we think that using these organoids, we can understand some of the primary defects that occur into human neurons. And so maybe to understand what could be the primary things that somehow are degenerate, and then that are the ones that maybe then are important to target. So then, of course, the disease might develop a bit later because of maybe inflammation or other effect, but maybe by using this model, we can understand something that happened very early. For example, like I mentioned, this ability to make branching. And then using this system, we are also hopefully able to identify some treatments that are maybe not curing completely the disease, but maybe are ameliorating some components. And then since we use FDA approved drugs or repurposable drugs, we hope that this can be quickly repositioned to treat Leigh syndrome families. And we are basically planning this. We're in the process of planning a clinical study, although it's quite challenging, and we can discuss this With that, I thank everybody and all of you for the attention, and of course, Puremiter ATP6 for the recent funding. That we're very excited to work with Michael and Steve on new therapeutic developments. Thank you very much.
Thank you so